This is Module 11. In this module, we'll be covering the principles of enzymes, which are biological catalysts, and some of the basics of metabolism. We're going to look at the metabolism of carbohydrates, fatty acids, and amino acids. First, let's talk about enzymes. Enzymes, as I mentioned, are biological catalysts, and catalysts are materials that speed up chemical reactions, but by definition are not themselves consumed during the course of the chemical reaction. Most of them are mainly globular proteins, and they're very, very specific. We talk about the specificity in terms of the choices of which molecules will work with those particular enzymes. Oftentimes there's one and only one type of molecule that will work with a given type of enzymes. Most enzymes have an ASE ending, which is based on their function. So for example, the particular enzyme that changes glucose into fructose is known as glucose isomerase. It's the enzyme that changes glucose to its isomer fructose. Some other terminology that we're going to use with respect to our study of enzymes, we use the term substrate to refer to the reactant that is bound to an enzyme. This is the very specific molecule, and it requires a specific part of the enzyme, which we call the active site. This is the part of the enzyme that the substrate binds to, and this is where all the action occurs. And then after the reaction occurs, the active site releases the substrate, which has now become the product. Some additional nomenclatures, as we mentioned, the names of enzymes refer to their functions. So for example, for oxidation and reduction reactions, there are oxidoreductases and dehydrogenases. Oxidoreductases and dehydrogenases refer to redox reactions, which you'll recall are oxidation and reduction. Transferases, as the names refer to, transfer one group from one place to another. Hydrolases add OH and H, or as they're combined, they're adding an equivalent of water, usually the OH to one place and the hydrogen to another. Ligases and synthases are names that are used to join different groups or different molecules together. So for example, one very important enzyme is sucrase, which catalyzes the hydrolysis or breakdown of sucrose. Sucrose, you'll remember, is a disaccharide, and so when sucrose is hydrolyzed, it breaks down into fructose and glucose units. So the product, the substrate in this reaction would be sucrose, and the products would be glucose and fructose. The way that enzymes work, as do all catalysts, are by lowering the energy barrier, lessening the activation energy necessary to get the reaction to occur. The overall process does not change the enzyme, and the enzyme should be regenerated each time. And so usually in a given chemical reactions, the substrate is present in a quantity much exceeding that of the enzyme. Only a small amount is needed because it will be regenerated. They do not change the overall reaction energy, just making the hill, the chemical hurdle or the energetic hurdle, a little bit smaller, so it's a little easier for the substrate to overcome it. And many enzymes can increase the reaction rate 1,000,000 to 1,000,000,000 times. Enzymes are also used for diagnostic purposes. Monitoring the level of lactate dehydrogenase is often done to check cellular function. This particular enzyme catalyzes the reaction that converts pyruvase into lactase. Enzymes are also used in the process known as fermentation. In fermentation, you choose a source of sucrose, some sort of sugar. This can be, for example, wine grapes. And the first enzyme, invertase, breaks it down. It hydrolyzes the sucrose into glucose and fructose. Then another enzyme converts those molecules, this enzyme is called zymase, into ethanol. So you've changed and fermented the wine, or specifically the sugar in the wine, into ethyl alcohol. How do enzymes work? There's a specific site we mentioned on the enzyme called the active site. The substrate molecule binds selectively to that site. And if there are two substrates, they both bind to that site, the chemical reaction occurs, they are then released. We call the different states here, when the substrate is connected to the enzyme, it's known as the enzyme substrate complex, ES. the enzyme product complex, EP, and then finally the enzyme plus the products. So this is a breakdown. Notice that each one of these steps is reversible. What types of factors influence the rates of chemical reactions? Well, usually if we're working with enzymes, we're working in a physiological framework, so we have to take into account that there are preferential pH ranges for activity, and that depends on what part of the body that they're occurring in. If we have, for example, a reaction occurring in the stomach, which has a profoundly acidic pH, an enzyme like pepsin, which degrades proteins in the stomach, operates best under those conditions, usually a pH of about 2. Whereas salivary amylase breaks down starches in your mouth with saliva. The pH of saliva is usually ever so mildly acidic, around pH 6 to 6.5, and begins breaking down starches. On the other hand, an enzyme like arginase, which occurs in the urea cycle, which we'll talk about in a little bit, removing ammonia, which is toxic in the body, has to occur at a fairly basic pH, usually around pH 10 or so. Outside of this range, we can't have operational enzymes because under those conditions, the proteins that comprise the enzymes will denature. Other factors that affect reaction rates: temperature. Normally, under basic conditions, increasing temperature increases reaction rate, although there's a limit. Most enzymes occur at biological temperature, usually about 37 degrees C, but there are a special category of enzymes that occur at higher temperatures, known as thermophilic enzymes. These are the sorts of things that happen in hot springs and so on, and they're unusual, but their activity prefers those higher temperatures. One of the other factors we have to consider when thinking of enzymes are the possibility that there may be inhibiting mechanisms. There are two major types of inhibition mechanisms for enzymes. One is called competitive inhibition, and the other is known as non-competitive inhibition. One involves the active state, that's competitive inhibition, and the other does not affect or involve the active state. Let's talk first about irreversible inhibition. One good example is if you destroy the protein that is the enzyme by acid-base conditions, by high temperature, by mechanical degradation, that's going to make sure that the enzyme doesn't work. There's a reversible type of inhibition that's called competitive inhibition. In this case, we're looking at situations where a competitor actually makes it to the active site preferentially over the substrate. This is usually a molecule very, very similar to the substrate and effectively blocks it. If you stream or overwhelm the active site with the competitor, you can prevent the active substrate from occurring. The other type is known as non-competitive inhibition, which does not involve the active site. Usually, the non-competitive inhibitor occupies an alternate site on the enzyme, sometimes changing the three-dimensional shape of the enzyme so it can no longer accept the substrate. One good example of competitive inhibition is when someone accidentally takes a molecule that is similar to ethyl alcohol. This could be methyl alcohol, this could be ethylene glycol, the material that's present in antifreeze. How would you treat someone, or perhaps an unfortunate animal who's been poisoned by ethylene glycol or antifreeze? What they do is supply the animal or the person with ethyl alcohol as a competitive inhibitor, with the idea that providing an excess of the alcohol, while it may intoxicate the patient, is preferential to having the toxic effects of ethylene glycol on board. What will happen is the body's enzymes will work on the ethyl alcohol preferentially, and that ethylene glycol will pass into the elimination system. As I mentioned, non-competitive inhibitors bind to a site other than the active site, known as an allosteric site, usually changing the geometry of the active site and then making it unsuitable for the substrate. There are different forms of enzyme regulations. Many enzymes are present in the body in inactive forms known as zymogens. They have extra peptide segments that are not usually present in the active enzyme, and they have to be taken away before the enzyme is activated. Another form of non-competitive inhibition is when metals, particularly heavy metals like lead or mercury, can bind to some of the active side chain residues, particularly those containing sulfur like cysteine, and can then lower the enzyme activity. Some enzymes need cofactors, additional molecules or ions for catalytic activity. For example, to make collagen, vitamin C is necessary, and the disease known as scurvy is caused by a vitamin C deficiency, which is necessary for collagen synthesis. Other types of cofactors include niacin, another name for nicotinic acid, and this particular molecule is important because it gets incorporated into NAD+, nicotinamide adenine dinucleotide, which we'll talk about in a second when we talk about metabolism. Metabolism is how we use our food to make energy. And there's two separate parts of metabolism that are important to consider. There's catabolism, which takes larger molecules and breaks them down into smaller ones. Think of taking a polysaccharide like starch, breaking it down to glucose, and then using the glucose to make even smaller molecules. Anabolism takes smaller molecules and builds them up to make larger molecules. Let's first talk about the catabolism of carbohydrates. There are a couple processes that we're going to consider in taking a molecule like glucose, which we would get from the starch in our diet, and the number of processes that go to ultimately liberating the energy and breaking primarily the carbon in glucose, ultimately down to carbon dioxide. We're going to talk about glycolysis, the materials in glycolysis that then enter into what's known as either the Krebs cycle or the citric acid cycle, and then ultimately the chemical energy gets regenerated and we regenerate ATP in the electron transport chain. First, glycolysis. Glycolysis has as its main substrate glucose. And these are the different steps where glucose, remember glucose is a 6-carbon molecule, has phosphate groups attached to it. We transform those materials and ultimately split the 6 carbons in the glucose into 2 separate pieces. In this particular case, the separate pieces are going to be a dihydrogen phosphate and another piece which gets assumed and ultimately transformed into a molecule called pyruvate. So what we're doing is breaking up the 6 carbons of glucose into 2 pieces, each of which have 3 carbons in them. The pyruvate then is the material used in the citric acid cycle. The citric acid cycle can also be known as the Krebs cycle or the TCA cycle. Pyruvate enters into this cycle. At the beginning of this cycle, the pyruvate, which is one of the end products of glycolysis, is transformed into acetyl-CoA and then enters into the citric acid cycle. In the citric acid cycle, which converts the acetyl-CoA to citrate, succinyl-CoA, and oxaloacetate, the main thrust of the citric acid cycle, which is an 8-step process, releases energies and carbon dioxide at various points in the cycle. One of the other things that pyruvate is important for is that it gets converted to lactate during a time of, for example, intense muscle stress, such as an extremely trying workout. This occurs with an enzyme called lactate dehydrogenase, so an elevated level of that would indicate some muscle stress. For fatty acids, the mechanism by which the fatty acids are broken down is known as beta oxidation. All of the naturally occurring fatty acids have an even number of carbon atoms, and what happens during beta oxidation is that it breaks down the fatty acids in successive cycles that each time remove 2 carbon units. In this case, those 2 carbon units exit as in the form of acetate. So here's our beta oxidation cycle where we're reducing a 20 carbon fatty acid into an 18 carbon fatty acid. Beta oxidation occurs after initial conversion of the fatty acid into the thioacetyl-CoA ester. And once again, each cycle removes 2 units of carbon, ultimately exiting as acetyl-CoA. Peptides present an unusual situation when considering how they must undergo catabolism. Peptides contain amino acids, and amino acids contain nitrogen atoms, and removing nitrogen is important. And the way this is done is very economical. The nitrogen is transferred to another molecule known as alpha-ketoglutarate using the enzyme called transaminase. And so what happens is the nitrogen group gets removed from the amino acid. It becomes part of the new amino acid, the glutarate, and then ultimately that nitrogen gets transferred once again, but there's only one enzyme necessary for all the different amino acids. The other product of the reaction is an additional molecule of alpha-ketoglutarate. So this particular process can recycle the alpha-ketoglutarate in each step. That's an important, very economical procedure. It involves the assistant molecule of NAD+, which produces an NADH and an additional proton in each case. What happens to the nitrogen that gets removed? Ultimately, as it gets removed from the glutamate amino acid, we need the urea cycle. And the idea here is to selectively remove the ammonium ions, which are toxic, for ultimate disposal in the kidneys. The last step in the process is the electron transport. This process, the electron transport chain, is a sequence of electron transfer which occur in a cascade between different materials, different molecules embedded in a membrane. The idea here is that the oxidation reduction steps apply the energy needed to convert ADP to ATP ultimately and regenerate and those electrons that are used up in the process. The chemical energy generated in mitochondria is the result of a protein gradient. Notice that the first step, which involves ATP synthase, converts ADP to ATP and releases a proton, which goes from the outer part of the membrane into the inner part of the membrane. In oxidative phosphorylation, the hydrogen ion produced here combines with oxygen ultimately to make water. And this is a way to regenerate energy. The overall process that occurs here, combined with glucose metabolism, produces that in oxygen, and without oxygen, none of this would happen. So, what we've talked about in Module 11, the difference between catabolism and anabolism, the glycolysis cycle by which glucose is broken down into pyruvate. Pyruvate enters the citric acid cycle via acetyl-CoA. Pyruvate can also be converted to lactate. Fatty acids are broken down via beta oxidation, and amino acids broken down and reutilized in transamination, which enter into the urea cycle. And the last item we talked about was regenerating those energy storage molecules using the electron transport chain.